Good afternoon. My name is Jim Head, the section president of the Planetary Sciences section of the American Geophysical Union, and I'd like to welcome you to the Carl Sagan Memorial Lecture. Carl Sagan was an intellectual giant in many fields, of course, and an inspiration to us all as a scientist, as a teacher, as an author, and as any of you who knew him know, as a human being. It's entirely fitting that this lecture, in his honor, be co-sponsored by the Planetary Society, which he co-founded, the Planetary Sciences section of the American Geophysical Union, which in fact he was a member and an officer, and of course the new Biogeosciences section of the American Geophysical Union, of which he certainly would have been a moving force and an active member had he lived to this day. So welcome to the Carl Sagan Memorial Lecture, and I'd like to now turn the podium over to Diane McKnight, the president of the Biogeosciences section of the American Geophysical Union, to introduce our honored speaker this year.
Again, welcome. I am very pleased to introduce Professor Joe Kirschvink from the California Institute of Technology. Like Carl Sagan, Joe has dared to put forward in his career a number of exciting and unconventional ideas, and these ideas have focused on the relationship between biological evolution and major events in Earth history and the relationship between the development of the Earth and development of other planets. He has then imaginatively applied his training in the study of geomagnetics and other areas of geosciences to rigorously and vigorously testing and evaluating these ideas. One exciting idea that Joe has put forward, or was the first to put forward, was the idea that at various times in Earth history, the Earth was entirely frozen over, resembling a large, giant snowball in space. Another example of exciting research that Joe has done has been examining the magnetofossils in meteorites from Mars as probably some of the most convincing evidence to date of the possibility of early life on Mars. And I'm glad to report that the idea that he will be discussing today is equally, if not more, exciting than these two ideas, and I think we're in for a great lecture. So the floor is yours, Joe.
Well, thank you, Diane. Thank you, Jim. And thanks to the Planetary Society. In the spirit of being a Carl Sagan lecture, I'll kind of treat the topic today in a light mode. I hope I can entertain as well as educate. And in the process, hopefully, we'll learn something interesting about ourselves. So I've changed the title a little bit today. I'd like to say Mars, Panspermia, and the Origin of Life. Where did it all begin? And first, I'd like to say that I am going to propose something a little bit novel and strange. In doing it publicly, it's far better to have a peer-reviewed manuscript, at least in press. And that has happened. The manuscript basically reporting this is in press for Paleontologica Electronica, and my co-author, Ben Weiss, is hopefully in the audience somewhere because he keeps me honest in planetary science things. I'd also like to acknowledge that this is being webcast. It'll be available on the AGU website, and I'm also planning to put the raw PowerPoint file that I'm using on my website as well. If anybody wants these figures and things, you're welcome to them. I'll probably see them hitting me in the head for years to come, but I'll do it anyway. And also I'd like to acknowledge the funding agencies, of course, that have driven our knowledge of Mars, in particular NASA Exobiology Program, the NASA Astrobiology Institute, and major sources of data that include many Martian space missions, not just NASA's but others, microbial genome sequencing projects. I'm actually going to depend a fair bit on some rather unexpected spin-offs from the Human Genome Projects, particularly the microbial projects for which we now have about 60 complete genomes. And finally, I'd like to acknowledge Hojatola Valley, who is actually the guy who first pushed the McCamick team into looking for bacterial magnetofossils. So to start the Sagan lecture, I'll start with something Carl Sagan did. And it's actually a quite amusing thing. Back in 1966, Carl and Soviet scientist Shklovsky published a little book called Intelligent Life in the Universe. Now, in this, they actually have a nice quote from Christianus Huygens, which goes, "Nor has Mars any moon to wait upon him, and in that he must be acknowledged inferior to the Earth." Well, of course, we now know that Mars does have 2 small satellites. Huygens didn't know that. He didn't have a good enough telescope, so, you know, the fact that he was wrong there can be excused. But chapter 26 in this book are the moons of Mars artificial satellites. I'd like to remind you that, you know, this is seriously proposed by Carl and Shklovsky, and they actually thought the Martian satellites, particularly Phobos, because it's in an unstable orbit, might actually be hollow and be filled with libraries and the relics of universities and things. But of course, you know, we now know that, no, they're pretty clearly primordial objects and trapped, and they're not artificial, and the fact that Phobos is in a decaying orbit is not unexpected. Well, in that view, preface my talk. So if I'm wrong, well, it's an honest attempt and it's fun, and maybe we'll learn why I'm wrong. So here's what I want to do. I'll talk first about the whole idea of panspermia, and then relate it to the early environment of Mars and the present Martian life debate. But then I'll focus on the same thing for Earth. What about the early environment of Earth and the early life on Earth debate, to see if there's any better evidence on Earth than there is on Mars for early life. And finally, the fourth part will be the ancestry of metabolism, and the question, can we use what we now know, particularly new results from the genome projects, to make a guess as to where life originally evolved? Yeah, please turn down the lights. That might be a little better. Thank you. The idea of panspermia actually goes back at least 2.5 millennia. First fragments of the idea were on surviving bits of the Greek scientist Anaxagoras, who envisioned life being scattered as seeds through the cosmos. By the way, the term panspermia actually predates the discovery of spermatozoa, and it is actually the proper scientific term for the concept of life moving around. We had one reviewer for our science paper last year who didn't quite get the hang of that. Anyway, it was discussed extensively in the 1850s onward. In fact, Gustav Arrhenius told me that his grandfather actually did a lot of work on panspermia. But prior to the Big Bang idea, the universe was thought to have had an infinite duration. If you have an infinite duration universe, the idea that life might eventually jump between planets is not a dumb idea. Rare events would eventually happen. So it's kind of a neat perspective. Today, you know, with the Big Bang and whatnot, we really don't think panspermia going between star systems, for example, would be very likely. And, you know, even more recently, Francis Crick and Leslie Orgel even suggested maybe there was directed panspermia, that you could overcome some of these problems with an intelligent civilization out there. Well, the bottom line is panspermia possible. And I'll go back to a quote from Svante Arrhenius back in 1908. "Fantastic is the idea that organisms caught in meteorites are carried out into universal space and deposited in other celestial bodies." And he goes on. Arrhenius' problem was, you know, the surface of the meteorite becomes incandescent with heat. It actually melts the surface. And he said, well, any germs on the surface would have been destroyed. Well, the ideas for panspermia have improved quite a bit now. We now know that there's bacteria in rocks, and they're not necessarily burned on the surface. I'd also like to thank the Planetary Society last November for publishing and commissioning a beautiful painting by Don Davis. It's basically showing the hypothetical early Earth being splashed by an impactor and, you know, putting the seeds of bacteria out into space. Well, we do now know that we have Martian meteorites. We have, I think, 19 at last count. And the early suggestion that these things were Martian actually goes way back. Don Elston, a paleomagnetist, in his PhD thesis at ASU, first suggested that Lafayette was probably Martian because of its magnetic properties, and Stolper and McSween came in later and refined that with the higher science of petrography. Anyway, but now we know that there are many lines of evidence that these are Martians, including the trapped gases, where in the Viking landers we know what the Mars atmosphere looks like, and the Shergotite glasses. The rare gases all plot very nicely on this. So there's really no big debate about whether these objects are Martian. And of course, the big problem is this rogue Martian meteorite. I'll be talking about it extensively in this talk. It has an interesting history. It is the oldest rock that we have from any differentiated planet. It was beaten up badly during the early part of the solar system, and by about 4 billion years ago, it got brecciated, something rotated fragments, didn't hit it too much. Carbonates were formed, and then there were other impact events, apparently. And then it sat basically doing nothing on Mars until about 15 million years ago. It was blasted off space. It landed in Antarctica about 11,000 years ago, and best guess is about 500 years ago it came to the surface in the Allen Hills of Antarctica. Well, we started work on this not really to verge into panspermia, but to see if we could use the remnant magnetism in the rock to say something about the Martian magnetic field. And after doing an initial study on 2 grains that we could reorient, we started collaborating with Franz Bodenbacher and group at Vanderbilt University using a wonderful new microscope. I show it here for the paleomagnetics community here. It's basically a very simple device. We have a superconducting Dewar. This is a conventional magnetometer used in most paleomagnetic laboratories. We put the sample inside the superconducting ring and you measure the flux there. But actually, by putting a pickup coil right next to it, you can take a room temperature sample right next to the 4-degree Kelvin pickup coil and intercept most of the flux. And you can actually raster and scan the system. Believe it or not, that device increases the sensitivity to small magnetic moments 4 orders of magnitude. It's incredible. We described that in an EOS article a couple of weeks ago, but it is really a major advance. And actually, the best samples are thin sections, which geologists use left and right. So it's a wonderful new instrument. And by the way, it will be available soon. The Tennessee group is arranging for it to be commercially marketed. And I've got to tell a story. When we first started working on this Martian meteorite, the only instrument that could do this work was, again, in Tennessee, and they do not have a paleomagnetics laboratory at Vanderbilt University. So we had to build a furnace that we could do the heating experiments to measure the Martian paleointensity in Pasadena. It's a 3-foot-long gizmo in a metal tube. And we had to, we said, well, we can't ship this to Tennessee because it's going to, end up in pieces. So we put it in a duffel bag and, headed for the airport. And we kind of got in line, gee, we have to go through a metal examiner, a metal detector, security check. So, well, we crossed our fingers and put it in and let it go through the X-ray machine. And we wandered through, and we had the security person had the funniest look on her face. Said, what is this? And we said, well, it's a piece of scientific equipment. Oh, I'll have to go get my supervisor. And so a few minutes later, this young college-educated gal comes out, and very proud that she has something to do finally. And she looked at this thing and said, all right, what is it? Well, it's a Tellier furnace for measuring the intensity of the Martian magnetic field. She looked at it and said, And of course, all the other security guards were watching her. And she said, well, I saw one of these last month. Go ahead. We haven't been able to bring it back since last September. Anyway, but with that, we're able to do standard paleomagnetic studies on the rock. And this is an example of one of the first things we did. This is the fusion crust of the Allen Hills meteorite where it came in through the atmosphere. You can see a very intense magnetic anomaly. In fact, Arrhenius was right. You know, certainly the surface of the meteorites become incandescent. They melt, and they also remagnetize. You can actually see that in the magnetic signature. But he was a little bit wrong because the heat pulse only goes a few millimeters into the rock. You can actually see a bleached zone here where it's been completely remagnetized. Well, deeper in the rock, the interior actually remains a very nice heterogeneous magnetization. You can actually see the actual instrument here is giving you the perpendicular component of the magnetic field, blue being in, red being out, and so you can actually see heterogeneous patterns in there. And when you actually go into the interior slices of this thing, you see kind of what I described as a magnetic battlefield, you know, blobs and craters and bits, dipoles actually pointing in all different directions. But, you know, since this is, you know, a Martian sample, we were going to do the magnetic analyses as gently as we could think of. And so we started the Tellier series, which is a series of heating and cooling steps in controlled fields at what we thought were ridiculously low values so that we would capture all of the data that we could from this precious sample. And so the first step we did was a room temperature scan, which you see here on your left, and you can see these nice dipoles. Then we heated it to 40 degrees, well, because, you know, 30 degrees was only 5 degrees up, and then 35 degrees it was only, well, anyway, so we did 40, and we did that, and suddenly got stunned at the difference. I mean, for example, there's a huge dipole here that's almost completely gone. A lot of these arrows show the matching things, and in fact, there's a lot of magnetism that is lost during those two steps. In fact, I'll show you a transect now between A and B here in the next slide, A to B. These are different transects going all the way up to 200. We haven't shown all the data, but you can see there's a huge drop just getting up to 40 degrees Celsius. And what, in fact, that told us was very exciting. It wasn't something we'd really planned to do, but 40 degrees Celsius is just a little bit above your body temperature. The human body is about 37. So 40, and that's an upper limit, actually. Most organisms can survive that. I mean, the inside of that meteorite, which remember is a 4.5 billion-year-old meteorite, at least since the magnetic pattern was formed in a heterogeneous fashion, had never been heated enough to do that. And we followed that up with a number of other experiments, and it's pretty clear that we can show that that heterogeneous magnetic pattern, those dipoles, go back at least to 4 billion years. And in fact, there are a number of ways of doing it, looking at argon loss and blocking temperature spectra and things. And it's very clear that that magnetic pattern goes back all the way 4 billion years. And what that means is kind of neat. We had a rock that had never reached 40 Celsius in 4 billion years, and yet it's sitting here on the table. And so when you combine that, and that's the first observation, it's a low temperature transfer. When you combine that with the European Space Agency results, they actually had a long-term exposure facility here where they put it in orbit for 5 years and put a whole slew of bacterial spores, even things like spinach seeds, in space, and discovered that, gee, space conditions are not, you know, that most things and many spores can actually survive this transit. And so here's the conclusion of part one in terms of the panspermia. A little bit more information. It's not heat sterilized. Milakovsky et al. In the group from the University of Arizona, Jay Malash and others, actually published a paper almost simultaneous with our science paper that, you know, billions of tons of Martian rock get transferred naturally. I mean, it's about a ton a year that comes in, and just dynamically every 100,000 years or so, which is geologically an instant, the rock goes from Mars to Earth in less than a year. And we also know from the ESA studies that bacterial spores survive in deep space for more than that. And we also know, again, from the University of Arizona group, that the bacteria can survive these accelerations and jerks certainly 10 times higher than that expected from the ejection process from the Martian surface. The bottom line is pretty clear. The transfer of life, bacteria, archaea, and even eukaryotes, is not just possible, it's a likely event. It's something that if those things were on Mars, they would definitely be here. There's some disagreement about whether you can go the other way. It's much more difficult to get off the Earth and to climb out of the solar gravitational field towards Mars. But the University of Arizona group estimates maybe a 50-to-1 ratio between transport the other way. 50 times more likely from Mars here, but they say it's not impossible the other way. But again, that needs to be tested. We don't have any meteorites from Venus, Mercury, or even re-entry meteorites from Earth that we can show were launched without heating. I mean, that's a big unknown. So anyway, it's a likely event. And so, you know, in terms of the origin of life debate, that makes a rather big change in things. We now have to worry not just early Earth, but early Mars. If life evolved in either place, we could be here today. And so we can't assume that it's on Earth, simply because the dynamics of the transfer process are so easy. So let's look at early Earth and early Mars and see what the current state of knowledge, such as it is, might tell us is going on between these places. Again, the last 2 or 3 years have been incredibly exciting for the AGU community in particular, particularly with the Mars Global Surveyor results showing that there's first beautiful magnetic anomalies on Mars. Mars is the bottom slide here. And if you look carefully between the magnetic field of the Earth, you'll see there's a factor of 10 increase in intensity. The Martian anomalies are incredibly strong. And it's been made in several smaller sessions, but to think about it, you could stand on the surface of Mars and your compass would work in most places. It wouldn't point north, but you know, the magnetic field is strong enough in most places. It's an Earth strength field at the surface. It decays away because it's from crustal anomalies. At the moment, there's not an internal dipole. But when you actually look at the anomalies, and this is a beautiful image from Connerty et al. In Science a couple of years ago, there are even magnetic stripes. Well, it turns out, because we know, the trouble is there's no data on those stripes. It's just satellite observations. But because we can actually date the magnetism in this ALH 8411 meteorite to 4 billion years, that's one dated constraint in the entire Martian magnetic history. And we know that those magnetic stripes, the dynamo in Mars, had to have turned on before, well before 4 billion years. Could have shut off before the rock was formed because the crustal anomalies are strong enough to magnetize it, but at least it's a constraint on the origin of the magnetic field, and that has life implications. So it's active prior to 4 billion years, and that means in the early Noachian part of the Martian history, there was a shield against solar sputtering and solar erosion of the Martian atmosphere. Not only that, if you stop the solar wind ablation, you disrupt the present 2-to-1 hydrogen to oxygen escape ratio, meaning that you've got the potential, at least, of oxidizing the Martian surface environment early on by simple thermal loss, Jeans escape. It works much better on Mars than it does on Earth, simply because Mars is a smaller planet. And it also is consistent with hydrogen to deuterium and oxygen isotope ratios. Also, I'd like to note that there is evidence, I'm not sure how strong it is, from Jim Farquhar et al., that there is a mass-independent oxygen fractionation from the Allen Hills carbonates that suggests the Martian atmosphere at 4 billion probably had an ozone stage, which is consistent with the idea of the atmospheric shield operating at that time. But that, in turn, also implies that early Mars would have had a UV screen, which is extremely important in virtually all scenarios for the origin of life, so that you don't have ultraviolet light disrupting prebiotic molecules. So those are very important things. We also have been very excited in the last few years by the discoveries of clear evidence, not just an occasional stream channel on Mars, but ocean basins. And I refer to Jim Head's work here, and possible shorelines even. And this is what I thought was one of the most beautiful things, the Phillips Zerber et al. Paper in Science earlier this year, showing that the Tharsis volcano itself warped the mantle, this already solidified mantle of Mars early in Noachian time. And in fact, this is Tharsis. It's the largest known gravity anomaly in the solar system. And in fact, if you look at that warping and then you look at the drainage channels, they're all downstream of that. That provides a constraint on when the huge volcanic complex of Tharsis came in. And it's actually quite nice because Tharsis is old, at least the bulk of it. Certainly there's younger volcanics on the surface, but the bulk of it's old. And that means you have a source of massive volcanic gases right when the magnetic field was shielding it. So it leads to, you know, a nice mechanism for suggesting you did have a nice atmosphere, maybe enough to warm the planet to the liquid water state, hundreds of meters to maybe several kilometer deep ocean basins. And you know, it leads to the question, if life wasn't there, why not? I mean, it's a place that certainly based on terrestrial analogs, it should have been. So that brings me to the next part of this Martian thing. I mean, I talked about the environment of early Mars. But what about the Mars life controversy? It's probably one of the most controversial areas in science. And so here I go out, like everybody else, on thin ice with people with little hand grenades quite willing to disrupt the ice sheet, okay? So how do you recognize or detect ancient life? I'm going to show a series of slides here. I'm going to flash them up pretty quickly, and I'm going to ask you, just as a general audience, which left or right screen is a better biomarker, something that if you saw waving at you from a Martian lander, you would be more convinced, okay? So here we go. Number one. In fact, I'm going to show it, and then if you think the one on your right is the best, raise your hand quickly. Okay, about half of you did it. And the other half said, well, there must be a trick here. And of course, here's the trick. Try the next one. I made up the name Phasianus marsicus. Anyway, and of course, it's the face on Mars, and the MGS certainly got much better resolution than the Viking landers or any of the other imagers had of it, and we now know it's not organic. So the bottom line here is you can't rely on simple anthropocentric analyses to decide what is living and what is not. It's a common mistake that you make, and a lot of paleontologists fall into that trap. So let's see if we can move away from a human-centric view and pretend we're intelligent jellyfish. Okay? If you're an intelligent jellyfish and I ask you to raise your left or right tentacle, you know, you would probably, well, all right, I don't know about this. We occasionally see these things roll down and fall into the seashore, and yeah, but yeah, we don't know what they are. But, you know, this looks like Uncle Harry, and you know, fine, we'll think that. But let's extend the analogy a little bit more. What if you were a slime mold? An intelligent slime mold, okay? And you were confronted with this, or, you know, bacterial magnetofossils, you know, which we've been studying in the paleomagnetics community for 20 years, and prior to the ALH 84001 debate, nobody really ever questioned that when you saw those in the TEM grid from a mud separate from the South Atlantic, that it was not biology. People have been trying to make them, and they couldn't do it. So the point is, you really have to get away from your Earth-centric view when you're trying to do it. And when the Astrobiology Institute sat down and had discussions about how to detect life and recognize it, it's clear there's an ideal goal that, you know, the one thing that distinguishes life from inorganic material is the result of Darwinian selection, natural selection, evolution through Darwinian evolution. But the problem is how to recognize it. I mean, it's ideal, but not always applicable. So here's the question, you know, like, is this really a Martian or is it something else? And the McKay paper was actually seminal, no matter whether you believe it or not. It's stimulated more thought of what to do and how to do it than anything else. It led to the National Astrobiology Institute even. And they had 4 lines of evidence, from organic materials to these worm-like things, mineral assemblages out of equilibrium, and at the very end, they said possible magnetofossils. Now, Valley had just joined the McKay team and didn't really have much time to contribute to that before it was published. But, you know, this paper it's actually followed up by Gibson et al. In pre-Cambrian research recently. The McKay et al. Team, by and large, has not given up on any of these observations as being biomarkers. And there's still a very vigorous debate going on it. But I'd like to compare the ALH life debate in a humorous way to the old Indian legend of the 6 blind men and the elephant. Okay, and I'll read this little poem after John Godfrey Sax's late 1800s poem. 'Twas 6 men of Indostan, to learning much inclined, who went to see the elephant, though all of them were blind, that each by observation might satisfy his mind. And of course, it goes on, and one sees the tusk and thinks it's a spear. The trunk looks like a snake, and the tree is the leg is like a tree, and so forth. And the conclusion is wonderful. And so these men of Indostan disputed loud and long, each in his own opinion exceeding stiff and strong, though each was partially in the right and all were in the wrong. Now, the elephant and ALH 84001. So we will modify our elephant a little bit and look at, organic contamination or biogenic pause, high T carbonates, low T carbonates, vapor phase magnetite, bacterial magnetite, shock-induced, it goes on and on. I could go more of this. But the bottom line is, how do we go down and recognize life? And ultimately, you've got to step back and realize that you're looking at an elephant, and you can't just, look at the tusk or a bit of the hide. You need to go back and actually look at the whole beast. And so the question then comes, what should you look for to prove the existence of past life on Mars from a meteorite? And again, just remind you, the McKay et al. Strategy was you get multiple lines of evidence, and although no single line of evidence was presented as individually compelling, collectively, McKay et al. Said this is the way to do it. And that has been incredibly severely criticized. Requiems for life on Mars have been published in Science and da-da-da-da. And many, many prominent scientists have said, in their papers, looking at any particular one of these things, saying, in any case, the attached figure establishes, insert whatever result they're talking about there, is not unique to biogenic processes. Okay? That phrase has been repeated over and over again in the last 5 years. So, let's ask the follow-up question. If whatever the result here was is not unique to biogenic processes, what is unique to biogenic processes? Okay? And guess what? Nothing. Absolutely, full stop, nothing. Why? It's dumb and simple. Life obeys the same physical and chemical laws that govern everything else in the universe. If you were to find something that life did that violated Newton's laws, the laws of gravitation, the laws of electrochemistry, what would you do? Would you be like the Bioelectromagnetic Society does, with people running around saying no KT on my t-shirt? No. What you do, you go and you look at the problem, and if you find indeed that you have an experiment that violates nature's laws, you change nature's laws. They're all empirical. And that, frankly, hasn't done. I mean, people used to think the bumblebee couldn't fly very well. Actually, nobody ever seriously proposed that. They said, look, we can't explain how a bumblebee flies, and what you do is you change your engineering and your physical understanding of it until you understand how it flies. The point is, it is extraordinarily unfair for critics of McKay et al. To say, I can do this inorganically, therefore you can't use it as a biomarker, because there's nothing you can do. And I'm not accusing the critics of the McKay et al. Paper of being creationists, but the logic they are taking, in the author, the previous slide was a direct quote, it reduces fundamentally to an untestable hypothesis, which is in principle no different than creationism. Okay, so what do we do? Well, anything else is magic along these lines. and I've already mentioned that. So, life recognition part 1. Identify a trait that is important to survival and fitness of the organism, something that Darwinian selection can use. And in the example that will follow will be the magnetic bacteria. The magnetic moment of these bacteria is very important. We know how it operates. It has single domain magnetite or sometimes Greigite that provide little dipole magnets, permanent bar magnets. Magnetosome formation is under genetic control, so it's a nice thing. You can actually sit in and manipulate it. And there's another thing. It's not just any biomarker. It's kind of summarized here as Andy Knowles' law. The value of a biomarker is directly proportional to the difficulty of making it inorganically. If you try to use a biomarker, if I could sneeze at a clay stone and make a human skull, the human skull wouldn't be a very good biomarker. Okay? But it's not easy to make. And it does show the fingerprint of natural selection, actually. So the idea is as follows, and the idea is that if you have something that varies like chemical purity that life can control, they're easy to make low purity things, and it's more difficult to make things pure. Particle size, it's easy to make it in some areas and not others. So the idea is that you go and use multiple parameters, and you try to find an intersection where life has driven things in various directions. And actually, that's not too dissimilar from what McKay et al. Said. They didn't put the fingerprint of natural selection in there, but they did recognize that the various biomarkers can be tilted one way or the other. So here's a magnetic bacterium. We're going to focus on this. It's actually a nice bug. We have a complete sequenced genome for it. The Department of Energy did that for us last November, about a year ago. And we've been slowly assembling the genome piecemeal. It's 4.3 megabaits. We've got restriction maps for it, and they're actually nice things. But the nice thing about the magnetic bacteria, it's one of the few biomarkers that we can really put Darwinian selection on it. We know how these bugs use the magnetic field. We know why they use it. We know the selective advantage. And so we can actually apply this test of a biomarker with it. And I've been studying these bugs, as Diane mentioned in the introduction, for over 20 years. And in fact, I actually discovered south-seeking ones. But it's pretty clear that they use the magnetic field to go up and down. It's their way of combating Brownian motion. The magnetic to thermal energy ratio for the geophysicist here is about 10 or more. So the Brownian dispersion is minimal, and these things are just swimming bar magnets, nice compass needles. And they use the magnetic field. And in fact, that's where the oxygen gradients are, and that's where they like to survive. And I'll give you an example, that a very simple high school level experiment that demonstrates that this is what in fact they are doing. This is called the Kelmine-Blakemore pulse remagnetization experiment. You start with a bacterium who wants to swim to the north, and all the little crystals are magnetized so that they're in one direction, as you can see there. You then hit it with a sharp magnetic pulse, anti-parallel to the direction, and that's shown here by the little lightning bolt. And bang, what happens is you can actually remagnetize each crystal. So this is pointing that way, this is pointing that way. The bacterium doesn't know you've done this. It's just an internal thing, bang. Its magnet is this way, and now it's that way. And what happens when you do that is our nice little bug rolls over merrily and swims the other way. And now I'm going to show a video clip here of this actually happening, but just to let you know what's going on. It is an unequivocal demonstration of single-domain ferromagnetism. It's rock magnetism and biology. And for the rock magnetist here, the pulse strength is set to be half of the average of the peak coercive force of the population. What that means is half of the magnetic bacteria will flip over and go the other way, and the other won't. And I'll show this here. Let's see. Go it like this. This is where I can pretend to be Carl Sagan and say, I have billions and billions of bacteria, and they're all doing my bidding. What I'm doing is, this is a pond water with Vaseline, and I'm moving the magnetic field left and right. These are north-seeking bacteria initially, so if they're swimming towards me, the north magnetic vector is pointing towards me. And when I turn the magnetic field around, the bacteria do a nice U-turn and swim back towards the Vaseline at the edge of the slide. Now, what I just did there was a pulse. I actually missed it, but you can actually see it shimmered, and now we have two waves, and they're crossing back and forth. Never the twain shall meet. I've remagnetized half of them. Okay? That is a unique ferromagnetic effect. It's a wonderful demonstration that these guys are really obligate on the magnetic field. Okay. So quickly, there's a lot of work that has been done in these bacterial magnetosomes. This is from a paper by Peter Busick. Beautiful imaging of the three-dimensional structure. In fact, I think you can even see some of the enzymes in the magnetosome membrane leaving dents here, but I'm not sure that's really the interpretation. We'll see if it holds up in future studies. We know that magnetosomes are not just in bacteria. They're in virtually all groups of eukaryotes that we found except plants and fungi, but certainly in animals all the way up to humans. And they actually form the magnetic sensory structure, and that's another whole exciting field of their use. We also know that the fossils that you can pull out of the crystal you pull out of the fossil record look very similar to ones in modern bugs. But more importantly, after the McKay et al. Paper came out, I mean, prior to that, nobody had ever questioned that magnetofossils were fossils, all right? But the level of certainty improved. I mean, incredible claims need incredible proof. And so after the McKay et al. Paper came out, I sat down with him and said, all right, now how do we actually test this in terms of biological fingerprints? And so far, we've come up with 7 things listed here, various things. I don't have time to go into each of them. But the point is, any one of these things you can do inorganically or organically, but it's the combination of which gives you an effective biosignature. And in fact, the interesting things that are in the middle, nobody has ever come up with an inorganic crystal that meets all of these, not even in the last year or two when people have been trying to do it. Single domain size and shape, this is for the rock magnetists, it's a well-known diagram for the magnetic bacteria. If the length and width of the crystals are just right, it'll be uniformly magnetized. You can actually see how some of these elongations mineralogically and at least topologically are, where you start with an octahedron and stretch it and open up 0, 1, 1 equivalent faces. You get these elongate things. There are all sorts of ways you can do this. Some of them are hard and some of them are difficult. And a more recent observation made by Kathy Thomas-Keptra is that a lot of the bacterial magnetites actually have these truncated ends, and that was an interesting observation. But in fact, we can't explain that in terms of a natural selection thing, because there are a lot of recent modeling that has been done in the rock magnetics community to actually see what the magnetic field, the internal demagnetizing field, looks like at various parts. And it turns out sharp edges tend to generate flower structures. And in those volumes of the magnetosomes, the magnetite is not uniformly magnetized. So the bacterium is much better off moving those atoms into the next crystal rather than putting them there. Anyway, it was kind of fun. This is my youngest son. I was sitting at home in Osaka playing with paper cutouts, trying to build a model of this, and he wandered over and said, "Oh, Dad, I can do that on the computer." And so he got Illustrator out and put together a little pretty cutout. I sent that to my group from Osaka, and I came back and found a couple of them hanging on the Christmas tree in the department. In fact, it was so nice, I'm posting it on my webpage at Caltech, and if anybody wants to print out an 8.5 by 11 sheet, it's there for making Christmas trees out of. You can tell them. Anyway, and we know how the magnetic bacteria organize these things. They actually have a lipid bilayer membrane inside the cell. It's matrix-mediated, precisely controlled mineralization. They set the size of the vacuole first, they infill it, and then let it mineralize, and that's how they control the size and shape of the actual particles. Okay, the rogue meteorite again. Carbonate blebs, I won't go into great detail. The big debate concerns the rims here, which are chock full of magnetite and pyrrhotite. And of course, the McKay group continued their studies, and Kathy Thomas et al. Published a number of papers looking at it. Now, when I first saw these dispersions, if they hadn't told me they were from Mars, I would have said, gee, it's a sample from the South Pacific. It looks like a dispersion of natural magnetites that you would get out of any deep sea mud. And in fact, a lot of the friends are there. Kathy, I won't go into great detail, actually recognizes 4 categories of different things, including things, about a quarter of which look very similar to biogenic magnetite. Well, you know, in a natural sample, you're going to have mixtures of everything that's in the environment. And since this is an igneous rock, it's not surprising to find a mixture. In fact, I gave a talk earlier this afternoon in the biomineralization section, where from actually rock magnetic techniques, we can detect about 20 to 50% biologic magnetites in those carbonates, just with rock magnetic techniques. I'm happy to discuss that with anyone. But when you actually look at the morphology of these things, they are stunningly like those of the modern bacteria, including elongate things. And of course, you know, that previous slide was indeed from Mars, and in fact, my youngest son's model is actually from that particular Martian elongate crystal. So, there is another thing. There's a paper by Alan Emery Friedman et al. Arguing that there are magnetosome chains, and in fact, when we did the rock magnetic test developed by Bruce Moskowitz, as I reported earlier today, yes, we get about 30 to 50% things that seem to be in relic chains. At any rate, if you believe, you know, exactly, none of this is uncontroversial, I'll state it up front, but at least it has 7 out of 7 of these characteristics. And really, the best inorganic magnetites meet only a few of them. Now, I'll compare this. It turns out that magnetite has been studied not just by the rock magnetists. It was the classic ferrite discovered and worked on 60 years ago when magnetic recording technology first came into being. It's been the a guinea pig for, you know, rock magnetism for years. And it turns out the populations of magnetite produced by these bacteria would make ideal recording media. In fact, the hard disk drive aspect of that industry alone is $35 billion a year. And they've been working for 60 years. And again, the best they've been able to get is 3 out of the 7 things. They can't optimize the size distribution in particular, which is what the previous, the background slide mentioned. You can't stop this log normal crystal growth problem. It's a biggie. And again, going back to Andy Knowles' law, you know, a good biomarker is difficult to make inorganically. You know, the conclusion here is early Mars would have been a good place for life, and we cannot disprove the hypothesis that those are biogenic magnetites. And we've been trying. You never prove something in science, you can only disprove it. And we have failed to disprove that. And I haven't seen anybody that has. So now we're about halfway through. We'll get by a little more quickly here. I'll go through the early environment of the Earth. And of course, we had a whole symposium on this yesterday. The classic ideas where we have rounded detrital pyrite and uranonites in the Archean sediments suggest that there was not much oxygen. Classic distributions of banded ironstones. This particular one here shows a goodie. The banded ironstone distribution goes up and down, peaks at about 2.5 billion years, plummets at about a little after 2, and there's a little trickle in the oldest sediments we have. But more intriguing, not only that, there's all sorts of people working on paleosols. They don't have time to review everything. But the bottom line is, it looks like prior to about 2.2 billion years, which is the Paleoproterozoic snowball, we had a reducing atmosphere on Earth. And that's going to be critical in the final analysis that I make, that this is a reducing atmosphere. In fact, the actual best constraint on the first oxygen, which I interpret as photosystem 2, is a weirdie. This is the Kalahari manganese field in South Africa. In order to precipitate manganese from solution requires molecular oxygen, full stop. And if you look at the time distribution of manganese, there is no sedimentary manganese prior to about the Kalahari manganese field. And that one deposit is 80% of the global reserves of manganese in one stop. And it turns out, guess what? That's right after the snowball Earth. This is the Oncolith Volcanics. Dave Evans and I had a paper a couple of years ago showing that this diamictite interfingering with these volcanics at an 11-degree paleolatitude. It's bang in the snowball zone, and that unit is right in the post-snowball aftermath. In fact, the way we think it formed, you cap the oceans for 70 million years, you build up all sorts of reducing iron and manganese. When it melts, it turns out the oceanic water composition is precisely what you need for cyanobacterial growth. We predict the whole ocean will turn into a green ocean, and you can easily get your 50-meter blanket of manganese everywhere. And in fact, my students keep telling me I should spend more time on this diagram, so I will. This is a takeoff of an earlier diagram from Jim Casting, where we show time before present in a billion years, and we're trying to get the oxygen level on the Earth's surface. The colored zones show kind of the range of estimates, maximum and minimum. If you have a methane greenhouse, as the session yesterday had heavy discussions, you're at 10 to the minus 8 bar of oxygen, almost nothing. The uraninite and pyrite survival, pictures I showed earlier, would be about here. But suddenly the snowball happens and you need massive oxygen. In fact, I think what happened is that photosystem 2 evolved right there. And it was so productive and so energy efficient, it just took over. And bang, probably brought down the methane greenhouse, causing the snowball, boom, and then afterwards, at least that's Casting's idea. And above this, I show geological constraints on things. The Martian dynamo was here, the banded ironstones are here, red beds there, eukaryotes and magnetofossils, there, da-di-da. So that's that. Now, on top of that, I've tried to superimpose what Mars should have been like. Again, we only have one data point, Mars today. You go back in time, Farquhar et al. I have this ozone measurement here, so it's somewhere up there, that red dot. And, if the Martian dynamo is there, then presumably the volcanic gases coming out would ultimately have been oxidized in the surface, giving us this redox stratified environment, kind of like the previous slide. And so there's a big difference between early Mars and early Earth, at least in terms of the general model. You probably throw eggs at that from all sides, but, it's a working model. Well, what about life on Earth? Okay. Oldest claimed life on Earth. We had a big debate about this yesterday in the Precambrian section. Steve Moses et al. Found, apatite grains in some of the Greenland supercrustals that had isotopic ratios suggesting isotopic fractionation by life. But of course, there's a debate raging back and forth. Sano et al. Actually dated the apatites. They're not 3.5 billion, they're 1.5 billion, so there's a problem there. But Gustav Arrhenius assures us there are still graphites and sediments that are not obviously associated with it, and this that are that light. So, it's not as clean a story as it was. But what's the next best thing? And the next oldest and best thing is for years has been considered the microfossils from the Warrawoona group in Western Australia, stromatolite things and filamentous-like organisms interpreted to be cyanobacteria by Sopf and others. But again, even that is under intense criticism. And in fact, Roger Buick has been one of the leading critics of those interpretations for years, and we had the opportunity to go with him in the field in Western Australia last summer, the Australian winter, our summer. But and in fact, they were able to relocate the Shop and Packer site from '87, from which all these beautiful fossils came from. And guess what? Roger's right. That bed cross-cuts the background. The background sediments sure have been dated at 3.5, but the actual samples of black cherts that have the microfossils are clearly in cross-cutting relationships. They're younger than the entire Warrawoona group. And you can see multiple generations of stuff coming in, being brecciated and turned in. You can even see a recent black chert vein that comes in. You see little splinters that go off and they form bedding. There's no way that things that are cross-cutting can be that old. So the bottom line is, it's hard. There are stromatolites, not carbonaceous but siliceous in the sediments. But, you can have stromatolite, you can have photosynthetic algomats without releasing oxygen. Photosystem 1 does not release oxygen, and there are whole varieties of other types that don't. You can even get algomats in the bottom of the Santa Barbara Basin today. Just having a stromatolite does not mean you're in the photic zone even. No evidence for cyanobacteria. And not only that, comparing it to Mars, I don't know of any $35 billion a year industry It has been trying to make stromatolites. All right? And so, summary and conclusion. I don't think evidence for life on early Mars is any weaker than that for life on early Earth. Maybe I'm a pessimist about Earth and an optimist about Mars. But yeah, I mean, so let's get to the conclusion of this thing. Ancestry and metabolism in the question, where did life evolve? I show this picture from a paper in Science that Eric Gaidos, Ken Nielsen, and I published a couple of years ago, which is a simple summary of reality. Life as we know it is electrochemistry. It's based on electrochemistry. And the basic way to read this is that you start with redox, oxidation, reduction couples that start here, they couple to something over here, and that gives you free energy change. That drives metabolism. Virtually every couple that might possibly lead to a proton being pumped across a membrane to store biological energy, if you go out in the environment and look for it, you can probably find a bug today that will exploit it. You can't couple this way from left to right going up, but downhill, it's free energy decrease, it's positive driving, okay? And so one of the things that I'm going to use to differentiate between Mars and Earth in terms of where I think life probably evolved is electrochemistry. And in fact, it's central. This is an example of what has long been known as a piece of something called oxidative phosphorylation. And your eyes are probably glazing over now. But the basic thing is there are enzyme complexes, particularly in the mitochondrial membrane in aerobic organisms, that shuttle electrons down an electrochemical gradient while they're pumping protons across the membrane. Now, they actually pump a proton across and put an electron this way. That causes a charge separation. It stores energy in an electric field. And there are other systems that then take that capacitive energy and generate adenosine triphosphate. In other words, the oxidative phosphorylation. Now, the interesting thing here, if you look, this oxygen that sneaks in at the very end reacts and forms water. That is called oxidative phosphorylation, the oxidative component. In fact, this terminal enzyme complex here is called the cytochrome oxidase because it's cytochrome C here that goes through and passes the electron to it, and it gets oxidized when it's frozen. That's what it's called. But it's actually at the terminal. Now, one of the important results from the genome sequencing projects, again, one of these completely unexpected things, is that when you look at the universal tree of life, we've seen many, I've been at many talks at this meeting where this tree pops up. We know where the root is. The origin was located here by gene duplication events, Manfred Eigen and among others. I mean, FATPases and elongation factors and all things that evolved and gene duplicated before the last common ancestor are there, so we know where that is. Well, it turns out with 60-odd genomes, whoops, we have a little bit of a clue about what that last common ancestor could do. And one of the things, it had 4 respiratory chains. I mean, in that pretty diagram I showed with all the different redox couples, it had chains terminating in oxygen, nitrate, sulfate, and sulfur. And in fact, the idea that terminal cytochrome oxidase was there is extremely well-founded. That particular guy was there. In fact, Russ Doolittle published a paper shortly after the Casnastra and Morena published their analysis a couple of years ago saying, this may be lateral gene transfer. That's one way of getting that enzyme in all 3 domains of life. But in fact, when you look at the cytochrome oxidase gene, it has the same topology as the small subunit ribosomal RNA. It's not lateral gene transfer. There is a small bit of one of the cytochromes that goes across a branch, but the terminal oxidase didn't. It was there. So our last common ancestor lived on a planet with redox gradients going down to molecular O2, wherever that was. It's in there. It's very clear. So and the question is, when was it, and where was it, and how was it? And I'll show here the result of a molecular tree, a recent one that Blair Hedges et al. Put together. This is based on as many different common proteins they could find, about 60 of them. And I like it more than some other ones because it actually agrees with the fossil record and the geological record. I mean, the error bars here for the various divergences. And if you look at, say, for example, the cyanobacteria, we actually have come on, wake up, there we go. We actually have lipid biomarker evidence of cyanobacteria at 3.5, which actually agrees very nicely with the molecular clock estimate of when that came in. And of course, the cyanobacteria doesn't mean photosystem 2 and oxygen. That's the Kalahari manganese field here. In fact, we know the cyanobacteria had to exist before photosystem 2 was moved into it. But anyway, it agrees very nicely. But you know, within that dating system, it's fine. The main thing, if you look at the estimate for the last common ancestor out here, basically the archaea versus eukarya, that is very clearly older than this. I mean, and that's the only important point I'm trying to make here. I don't care how older it is, but it's clearly older. And the last common answer is out there. So I'm going to compare this now with the previous slide of the Earth and Mars history. And I'll bring a little box in. We'll try to bring a box in. Come on, there we go. And this is now the Earth history box showing where that last common ancestor and the terminal cytochrome oxidase that was breathing oxygen is located. And it's right back there. Now, between the two of them, you know, gee, Mars certainly looks like a better candidate for a planet with enough oxygen, not just oxygen, but oxygen enough for this system to breathe. The bug was breathing oxygen, not just sitting there. If it's just in the environment as a poison, you would expect things like superoxide dismutases to be there. And of course they're there, but you wouldn't expect a system that couples down to it for metabolic energy. And in fact, you can even see how respiratory chains might evolve. You know, if you have a smothered planet like Europa or some interstellar planet or something, you don't have much of a geochemical gradient, and you might, if you're lucky, get some limited electron transport to go. And you could duplicate, you know, if the environmental gradient increases, you can get chain duplication events and extend the chain like that. You know, but the problem is this initial system has got to be really efficient from the start. If you're going to propose a biosphere on Europa on a very, very weakly energetic methanogenic couple of some sort, you have to explain how a system could evolve de novo to exploit a minute amount of energy. Evolution doesn't like to do that. Evolution prefers to take a sloppy system and make it better. And that's shown in this slide. If you have an electron transport chain, primitive one, where you go from one enzyme complex, bang, down to something else, you can be sloppy if there's a lot of energy around. With time, evolution can run that. You can get gene duplication events and improve the efficiency and fill in the rest. Now, I think that's a much more likely environment to get started. But of course, the people dealing with the origin of life, from Urey on in, they said, "Oh, we can't get prebiotic molecules if there's oxygen around." Sure, fair enough. But in any plausible geological environment, you get redox stratification, just like we have today. The oxygen is coming in from the atmosphere. You'll have a redox cascade going down. So you can get your organic molecules in deep ponds and basins with hydrothermal vents, build up the prebiotic molecules, and let them diffuse into the oxic zones, or vice versa. And that way, you can actually start with the whole range and then get it to go more precisely. So to wind up, where did life evolve? Various possibilities: Earth, Mars, Europa, and interstellar planets. Dave Stevenson, my colleague, suggested that a while ago. All have water, no problem. All organics, probably. Redox gradients. Earth, if you believe the geologic record, maybe. I mean large redox gradients, allowing things to breathe oxygen. Certainly Mars, yes, maybe that should be probably. Europa, we would argue no, and we argued in our science paper a couple of years ago. Interstellar planets, Dave Stevenson may have a different answer there, but I would say no. UV screen, Earth. If you look at the Pavlov, Browning casting paper, and even Rob Rice's poster yesterday, arguing very strongly, no, you would not get a good UV stream in a methane-based atmosphere. So the bottom line is Mars. And so it's not an impossibility that Darwin's warm little pond could have been a cold little puddle on Mars. And there's actually an almost proof of this. All right? These are the Yamamba girls in Tokyo. They're Martian. And so we salute you, all you descendants of space-traveling microbes from the red planet. Billions and billions of you. Thank you very much.